In the nitrogen cycle, the first one is nitrogen fixation. Here, nitrogen fixation, the conversion of molecular nitrogen present in the atmosphere into the available form of nitrogen. Be nitrate, nitrite or ammonium ions to the plants is called as a nitrogen fixation. So here there are two types of nitrogen fixations are there. One is called as physical nitrogen fixation, or we can call it as a biological. And the other one is called as a biological nitrogen fixation. So let us start with the first one, physical nitrogen fixation. So here this is a process where the biological system is not involved, or the living organisms are not involved. So this will occur in the atmosphere. So here in this one, sometimes we know that the thunders or thunderstorms will come, or the lightnings will come in the atmosphere. So the thunderstorms and the lightnings will release some energy. So that energy is utilized for the conversion of the molecular nitrogen into the nitrates. So this process which occur in the nature, so by physical process, is called as a physical nitrogen fixation. So whenever the thunderstorms and lightnings will occur in the atmosphere, the nitrogen which is present in the atmosphere will combine with oxygen and it will be converted into nitric oxide. The molecular nitrogen reacts with oxygen in the whenever the lightning and thunderstorms will occur, converted into the nitric oxide. This nitric oxide will further undergoes oxidation to form nitrogen oxide, that is nitrogen dioxide. The so formed nitrogen dioxide, so whenever the rain falls or some moisture is available in the atmosphere, that will react with this nitrogen dioxide and will come in the form of acid rains. So that will be shown here, that is two molecules of nitrogen dioxide will react with the water that is available in the form of moisture or the rainfall, and it will be converted into, when nitrogen dioxide will react with the water which is available in the atmosphere, it will be converted into HNO2 and HNO3. So in this process, the nitrous acid or nitric acid will be formed. This will come to the soil in the form of liquid, so along with this nitrogen, and it will react with the various types of the radicals which are present in the soil and converted into that particular nitrate molecule. So that is HNO3. If it is reacting with calcium or potassium salts which are present in the soil, and they will be converted into either calcium nitrate or potassium nitrates. So this is a way how this available form of nitrogen will be there in the soil. So this is a process of physical nitrogen fixation which occurs in the atmosphere without the utilization of any biological system. And here this type of the abiological nitrogen fixation or the physical nitrogen fixation can be done in a large scale in the Haber-Bosch process at 450 degrees of centigrade. So that is a way how this and by using 100 bars of pressure. So that is a way how here the physical nitrogen fixation can be done in the industries also. Next one, the biological nitrogen fixation. In the biological nitrogen fixation, different types of the living organisms are involved. So here the living organisms are the one which have the ability to convert the dinitrogen or atmospheric nitrogen which is present in the atmosphere into the available form of nitrogen. Here various types of organisms like bacteria and some blue-green algae, that is cyanobacteria, they have the ability to convert the molecular nitrogen into the available form of nitrogen to the plants. So here why they have the ability? Because one thing that bacteria and blue-green algae are considered to be the primitive plants or primitive organisms. So despite of that primitive nature, how could they have the ability to convert the nitrogen into the nitrates or ammonia molecules? So that is because of an enzyme called as a nitrogenase enzyme. So that is the one which is responsible for converting the which will provide the ability to convert the molecular nitrogen into nitrates or ammonium molecules. So here different types of living organisms are involved. So based on their nature, so we can say some of them are asymbiotic organisms and some of them are the symbiotic organisms, or we can asymbiotic nitrogen fixation. So let us see the nature of those organisms which are involved here. One, free living and aerobic bacteria which are involved in this process, like Azotobacter, Biserinckia are some of the examples of the free living aerobic bacteria. In the same way, some of them are free living but anaerobic bacteria. Examples like Clostridium and Bacillus species. So these are some of the examples of free living but anaerobic bacteria, and some of them are free living and photosynthetic bacteria. The examples of this type of bacteria are, one is Rhodospirillum, the other examples are Chlorobium, Rhodopseudomonas. So these are some of the examples of free living and photosynthetic bacteria, and some other are free living cyanobacteria like Anabina, nostoc. So these are various types of examples of the non-symbiotic or asymbiotic nitrogen fixers and their nature. Then let us come to some of the examples of symbiotic nitrogen fixers or symbiotic nitrogen fixation. So here, what is the difference between these two? Here in the asymbiotic nitrogen fixation, the nitrogen fixer is not associated with the host organism, but it is present in the soil freely. But here in the symbiotic nitrogen fixation, the nitrogen fixer will get associated with the host, and both of them are showing mutual beneficial association. In that process, they start fixing the nitrogen which is present in the atmosphere. So let us take some of the examples of the symbiotic nitrogen fixation, like examples of the plant like Cycas. So Cycas is a gymnosperm plant, and this plant will have special type of roots called as a coralloid roots. So coralloid roots are one of the example for nitrogen fixing roots. Why? Because so these roots contain two types of cyanobacteria. Those are Anabina and nostoc. So already we had discussed these two names in the free living cyanobacteria also, but the thing is the species are different. So different types of Anabina species and different types of nostoc species are involved here. Then if you take the leaf of the Azolla plant, so Azolla is an pteridophyte plant or fern plant. Here Anabina azollae is a cyanobacteria which is associated with the leaves of Azolla plant, which will help in nitrogen fixation. And if you take the example of Anthoceros, which is an bryophyta, so here also some species of like nostoc are involved in the symbiotic association. Likewise, if you take the examples, there are many such types. Okay, in the same way, let us take some legume plants. So where Rhizobium bacterium is symbiotically associated with the roots of legume plants, and they also form a special structure called as root nodules. So inside the root nodules, the rhizobium will have its habitat and start converting the molecular nitrogen into available form of nitrogen. Likewise, by forming the symbiotic association also, there are many such examples which have the ability to convert the nitrogen into the available form of nitrogen, that is nitrate, nitrite and ammonium molecules. So here let us see the biological nitrogen fixation in the plants. The method or mechanism of biological nitrogen fixation, particularly in the leguminous plants. So there are many such legume plants which have the ability or which form the symbiotic association with some of the bacteria, and they are concerned with the biological nitrogen fixation. So such as pea, clover, alpha plant, beans, lupinus, etc. These are some of the plants which have the ability to form the symbiotic association and helping the biological nitrogen fixation. These are been forming the symbiotic association with different types of prokaryotic organisms like bacteria, and they form special structures. In that process, what they will do is they will convert the molecular nitrogen into the available form of nitrogen, that may be nitrate or ammonium molecules. So in this process, how this the prokaryotic or primitive organisms have the ability means they have the dinitrogenase or nitrogenase enzyme. So here dinitrogenase or the nitrogenase enzyme, this is the enzyme which is present in the prokaryotic organisms. So this have two parts. One is called as a iron protein part, which is an dimer, and the other one is a molybdenum iron protein part, which is an tetramer. So these are the two parts which are being present in the dinitrogen, so which is helping in the nitrogen fixation. So this dinitrogenase is a enzyme which is responsible for all this mechanism to be done inside the prokaryotic organisms during symbiotic association. So here the entire process of this nitrogen fixation by in the legume plants is given in the form of equation, where two molecules of nitrogen by taking 8 electrons and utilizing 8 protons, and they need some energy also in the form of ATP. 16 molecules of ATP are required, and they form into 2 molecules of ammonia, and hydrogen molecule will be released. So here the energy which is present here will be, or we can say that ATP will undergo splitting and forms into and release some energy and forms into 16 molecules of ADP and inorganic phosphates. So here these are also of 16 molecules. So likewise here, this is the overall equation showing the biological nitrogen fixation in the leguminous plants. So here one molecule of nitrogen is reduced to 2 molecules of ammonia by utilizing 16 ATP molecules, and here in this process 8 electrons and 8 protons are also being utilized. So this is the overall equation showing the biological nitrogen fixation in the leguminous plants. Let us see some more examples of the nitrogen fixing microorganisms. So here those are some of them are forming some special structures. Those are called symbiotic structures. So here the name of the microorganism, the symbiotic structure which have been formed by it, and the host in which it is taking shelter. So let us take first some of the examples of the bacteria, the well-known bacteria, that is Rhizobium species. Different species of Rhizobium will form symbiotic structures with different types of plants. That may be leguminous plant, or sometimes some of them are non-leguminous plants also. So which type of a symbiotic structure that is formed by it is a root nodule, and the host may be the legume plants. Let us take any legume plant in which rhizobium will get associated with it to form root nodules. Then some other species of rhizobium will also form the root nodules, but it is now the root nodules are not being formed in the legume plants, but it is in the non-legume plants like Parasponia. Then that is, some species of rhizobium are associated with the legumes and some species are associated with the non-legumes also. But the type of symbiotic structure that is formed will be similar structures called as a root nodules. Then the second group are actinomycetes. So in this actinomycetes, one is a Frankia species. So this Frankia species also forms the root nodules, and it is taking the shelter on the host like Alnus, Casuarina, and Myrica like plants. So this is about the actinomycetes member. Then some of the cyanobacteria, let us see. Here also the same thing, that some cyanobacteria species will form association with different types of plants and helping in the nitrogen fixation. Let us take examples of Anabina and Nostoc. They form lichens, and here they have been associated with the fungi, and this fungi will belongs to either Ascomycetes and sometimes to Basidiomycetes also. This is one symbiotic structure. Then other one are Nostoc, which will form a structure with a thallus of a bryophyta organism called as Anthoceros. In the same way, some species of Anabina and Nostoc will form some special structures called as coralloid roots in a gymnosperm plant like in Cycas. The same way, Anabina azole. So this Anabina azole is associated with the leaves in a pteridophyte member, that is fern plant, aquatic fern, that is a azolla plant, and some members of nostoc, like one of the species of nostoc will form the stem glands in angiosperms like in Gunnera. Likewise, these are some of the examples of biological nitrogen fixers and their structures which have been formed by them and helping in the nitrogen fixation. Okay. In the biological nitrogen fixation, let us see the root nodule formation. Formation of root nodules in the leguminous plants. The soil contains the soil bacteria like rhizobium is an soil nitrogen fixing bacteria first. This is present in the rhizosphere, that is the atmosphere around the root, and here the different strains are there. So, the actual strains which are concerned with the nitrogen fixation have to be attracted towards the root. So, in this process then only the next step, that is the formation of nodule, will be possible. So, in the initial step, so then some secretions are being done by both the root of the legume plant and also the bacteria to form an association leading to the formation of the root nodules. In this process, first the root of the legume plant will secrete sugars, amino acids, etc. These sugars and amino acids which are secreted by the root of legume plants will attract the rhizobium strains. So, they are attracted towards the root, and here in this process, so the bacteria will also secrete one structure called as a nod factor. So, this nod factor which is secreted by the bacteria is responsible for curling of the root hair, curling of the root hair, and here the specific strains of the bacteria are being attracted because of a special protein called lectins. The specific strains of this rhizobium will be attracted towards the so that they form some sort of a binding with the root hairs of leguminous plants. So, that is because of the secretion of a special proteins called as lectins by the roots of leguminous plants. So, in this process, let us see it in the form of diagrammatic representation, where this is a section of the root which the root hairs are present. So, this is epidermal part of the root, and some of the epidermal cells are producing the hairs. So, we call them as a root hairs, and this is the inner part of the root. Here outside this one, so this contains the soil particles, that is the soil atmosphere will be present. This rhizosphere or the soil particles will contains the different types of the bacteria, but as we said that by secretion of the sugars and amino acids and the lectins, so the some sort of binding will be formed between the bacteria and the root hair. So, at the same time, what happens because of the accumulation of this bacteria near to the root hair, so all these rhizobium bacteria will be accumulated. Because of this one, what happens is the root hair will start curling. So, this is internal tissue. Can see here the root hair is showing the curling, and which is surrounded by the rhizobium bacteria. So, here the rhizobium bacteria will secrete a nod factor which is helping in useful for curling of this root hair. And after curling, so here gradually the cell wall will be ruptured, or it have been dissolved by the secretions of the bacteria. So that what we call they will secrete cellulase and pectinase. So here cellulase is the one which will digest the cell wall, cellulose which is present in the cell wall, and pectinase will digest the pectin which is present in the cell wall of the root hair. So this is secreted by the bacteria. So a small perforation will be formed at the tip, and here after that the next limit is the plasma membrane. So here the plasma membrane which have been formed by the vesicular structures of the Golgi structure. So it forms an invagination like structure. So that invagination like structure which have been formed. So this is a leaf curve, and here what happens, the cell wall will start invaginating towards inner side, and this invagination which have been formed by the plasma membrane will pass throughout the length of the root hair, and gradually it will enters into the inner tissue. So this is the inner tissue. So this thread-like structure which have been formed and invaginated entering into inner tissue, we call this as a infection thread. So this infection thread is formed by the invagination of the plasma membrane, and now this shows a passage for the bacteria. By the meantime, what happens is the number of bacteria which are present around this root hair will multiply, and their number will gradually increases, and all these bacteria will gradually pass into this infection thread, and they will gradually moving into the inner tissue along with the infection thread. So likewise they will reach into the cortical tissue, they will enter into the cortical tissue of the root, and when they enter into this one, so by that stimulation, the cells of this cortical will start stimulating and they will undergo division. So when they divide, what happens, the number of cells will increases, and that will be coming out in the form of a nodule like structure. So likewise from that cortex tissue, the nodule will be formed, and all these bacteria which have been entered into this inner cortex, so that they gradually increase their number there also by dividing, and majority of them will enter into the nodular structure which is coming out of the cortical tissue. So here all the bacteria which have been entered into this one, so those are called as bacteroids. All the bacteria now which are present in the nodule are called as bacteroids, and these bacteroids are surrounded by a membrane called as a peribacteroid membrane. So here from outside the nodule will appears to be like a brown colored swollen structure which is coming out from the root. So this type of structure is called as a root nodule or matured nodule, which is brownish and contains many cells. So these are the changes which are taking place during the formation of the root nodule. And here the nodule is having connection with the vascular tissue of the root. So the nutrient supply will be maintained, which is required for the bacteroids which are present inside the root nodule. So that is the way the vascular supply is also been there established between the root nodule and the root of the legume plants. The internal structure of the nodule, so it contains the membranous structure, this is a peribacteroid membrane, so this is the host cell, then inside which we can see the this type of structures, these are called as a bacteroids, inside which so, and all this is a cytoplasm of the host cell. So, this is the internal structure of host cell, which is showing the presence of bacteroids inside it. This is a host cytoplasm. This is a bacteroid, or we can say all these are the bacteroids, and this is a peribacteroid membrane. And near to this peribacteroid membrane, we can see some special type of pigments. Those are called as a leg hemoglobin. Along with the dinitrogenase enzyme, which is concerned with the nitrogen fixation. So, here which is present in the Rhizobium bacterium, so here it is a oxygen sensitive, that is when the concentration of oxygen is more, so the nitrogenase will become inactive, but in absence of nitrogen, the rhizobium cannot perform respiration, so that the concentration of the oxygen we have to be maintained in such a manner that it should not cause any damage to the activity of the dinitrogenase. So, that function is done by leg hemoglobin. So, it is called as a oxygen scavenger. So, which is a pink color pigment, which is similar to the red color pigment which is present in the blood, that is hemoglobin. That is why it is being called as hemoglobin, and it is present in legume plants. That is why it is called as leg hemoglobin pigment, which is acting as an oxygen scavenger and protecting the dinitrogenase enzyme from the toxic effect of oxygen. Next, mechanism of nitrogen fixation. In this process of mechanism, here the nitrogen is gradually undergoing the reduction, and by adding hydrogen molecules. So, in this process the atmospheric nitrogen is reduced to ammonia molecule. So, here for this process of reduction of the atmospheric nitrogen, they require energy, electrons, and also the proton molecules. Let us come to the schematic representation how this nitrogenase is helping in the conversion of or reduction of molecular nitrogen into ammonia. Here this is a schematic diagram of nitrogenase enzyme. And here the dinitrogen molecule will be added to this nitrogenase enzyme, so that first it will occupy the sites which are present on the nitrogenase enzyme, and so it can be represented like this. The two molecules of the atmospheric nitrogen will occupy the sites which are present on the enzyme molecule, that is dinitrogenase or nitrogenase enzyme. Here nitrogen is a substrate, and this is an enzyme. Okay, the substrate is binded to the active sites which are present on the enzyme molecule, that is binding of substrate to the nitrogenase enzyme. Now the process of reduction, every step the hydrogen molecules will be added to this nitrogen molecule. So here the first step of the reduction, where hydrogen molecules are added, two molecules will be added here. and gradually leading to the formation of this type of structure. In this first, one hydrogen will get binded to nitrogen and the other hydrogen will get binded to nitrogen molecule. So, in this process gradually the three strong bonds which are present between the two nitrogen will be gradually broken, and in place of the broken bonds the new bonds have been formed by the hydrogen. So, the next step of reduction, the further reduction will be taking place. That means two molecules of hydrogen will be added here, and that can be shown like this. The nitrogen space and already it have been formed bonding with the each of one hydrogen molecule. Now they form bonds with the two of them like this. Then the further reduction will be taking place. Where two more molecules of hydrogen will be added. Nitrogen, and here they form bonds. Three hydrogen have been formed bonding with the nitrogen, so that the three strong bonds are completely broken by this process. And here this is a way how this complete reduction of the nitrogen is taking place by addition of a total of 6 hydrogens to the two nitrogen molecules. So, in this process, in the final step, so what happens is the enzyme will be separated. So, that means the enzyme is separated, and it may further participate in the other process of nitrogen fixation. And here the substrate which have been taken here, the nitrogen molecule now will be converted into the product, and it is being released in the form of two molecules of NH3. So, this is a way how these two products will be formed. And here once again, whatever the enzyme which have been entered, so the same type of structural enzyme will be separated from this process. So, this is a schematic representation how the nitrogenase is concerned with the formation of the two molecules of ammonia from one nitrogen molecule. So, in this process the enzyme is having some specific role to be played in conversion of this molecular nitrogen into ammonia molecule. So, for the process of reduction of nitrogen into ammonia, they require some reducing powers like ATP, electrons and protons. All these are acting as reducing powers in which they are helping in the conversion of the molecular nitrogen into ammonia molecule. So, here the dinitrogenase enzyme which is concerned with the process of the nitrogen fixation. So, I already discussed like they contains the protein part, that is two protein parts are there, one is iron protein part and other is the molybdenum iron protein part. So, here in this process the leg hemoglobin is acting as a scavenger of oxygen, but in cyanobacteria where this leg hemoglobin is not present. So, particularly if you take heterocystus cyanobacteria, in this one they have special cells called as heterocysts. So, these will take up the function of leg hemoglobin, that is they are acting as the structures which will help in protecting the nitrogenase enzyme from the high concentration of oxygen. So, these are heterocysts are the one which are acting or taking up the function of leg hemoglobin of legume plants. Then here they require the energy, they require the electrons and protons. From where what is the source of all these electrons, protons and energy for the nitrogen fixation, means that is the respiration is the one which is providing this all these electron, protons and ATP which is required for the nitrogen fixation. So, here that is the pyruvic acid which have been formed in the process will undergo phosphoroclastic cleavage. In presence of pyruvic dehydrogenase enzyme. In the presence of pyruvic dehydrogenase enzyme, and it will be converted into acetyl phosphate and carbon dioxide. In this process, some amount of energy will be released. So, this energy will be, that is in the form of ATP. So, this will be utilized for this mechanism of nitrogen fixation by the plants. So, likewise here the dinitrogenase is taking or utilizing the energy, and here the energy will be entering first into the iron protein part. Here if you see the organization of the structure of nitrogenase or dinitrogenase enzyme. So, this is a iron protein part, and this is a molybdenum iron protein part. As a result of phosphoroclastic cleavage, so what happens is energy will be released. That energy is, that is ATP, will be taken by the ferredoxin molecule. So, in this process the ferredoxin will be, the reduced form will be converted into oxidized form. So, here when the ferredoxin is taking the electrons from that electrons from the phosphoroclastic cleavage of pyruvic acid, it will be undergoing reduction. Now, this reduced form of the ferredoxin will provide those electrons to the iron protein part. So, then the ferredoxin is converted into oxidized form of ferredoxin. Here the iron protein part which is been in the oxidized form, when it is it receives the electrons from the reduced form of ferredoxin, it will undergo reduction. So, that is converted reduced form like this, and the electrons which have been taken from the ferredoxin by the iron molecule, it is undergoing reduction, and this reduced form of iron will provide those electrons to the oxidized form of MOFE protein part. So, this is already in the oxidized form. When it receives the electrons from this reduced form of iron, it will be converted into reduced form, and finally these electrons have been received by the N2 molecule. So, that will be converted into ammonia. So, actually here the three things we have to consider. One is ATP, electrons and protons. So, electrons are moving through the iron protein and iron protein part and finally received by the nitrogen, but the protons are directly being taken by the nitrogen, N2 plus H2. Then here for this transfer of electrons through this iron and molybdenum iron protein part, they require some energy which is provided by the 16 molecules of ATP, where they are converted into 16 molecules of ADP plus 16 molecules of inorganic phosphate. So, finally 2 molecules of ammonia will be formed along with some hydrogen molecules that have been released. So, for this process they require a total of 8 hydrogen or 8 proton are required, and 8 electrons will be required. For this 16 ATP will be utilized. So, the entire reaction will be N2, 1 moles of N2 plus 8 moles of electrons and 8 protons plus 16 ATP converted into 2 moles of ammonia, hydrogen along with 16 molecules of ADP and 16 molecules of inorganic phosphate. So, this is overall reaction related to the mechanism of nitrogen fixation where the dinitrogen is participating in this process.